Okay, hello everyone. I'm glad to have this opportunity to participate in this symposium. My name is Yang Liu from China Agricultural University. So today I will talk about some works we have done on pomegranate in the last several years in Israel. So first of all, pomegranate is a very special kind of fruit tree because in the taxonomy, there's only one species in this one genus. And the pomegranate have native from Central Asia and have been disappeared to almost all over the world. And it have been an orchard crop for thousands of years and have a long history in many methodology of many cultures like Greece, China, also in Israel. So we can find here is two beautiful pomegranate fruit from the local of ARO, which present one of the 7 crops mentioned in the Bible. So pomegranate fruit and juice are well known for their medical and therapeutical properties. So they contribute to human health. So they can prevent a lot of different kind of disease, such as cancer. So in the last 2 decades, customers have become more and more interested of this fruit. And consequently, a rapid increase of demand has led a sharp increase of intensive orchard area. India, China, Iran, Turkey, and the US had like 3/4 of the world planting area. And in Israel, we have around like 2,500 hectares of pomegranate orchard. And the rapidly expansion of this crop volume and created several fundamental knowledge gap in its management. There's a lot of studies on pomegranate, but many of them is about the post-harvest storage or disease. And there's few studies on the nutrient management and the lack of the very detailed studies. So we want to wire the soil culture and fertigation system to evaluate the response of like tree growth, flowering, yield, and fruit juice quality of pomegranate trees to different level of nitrogen application in an accurate and accelerated way. So the experiment is conducted at Gilad Research Center of ARO in Israel, and the 2 years old of a wonderful pomegranate plant in the July of 2015. And they grow in 500 liters containers filled with perlite growth media. And we set at 8 different nitrogen level from 5 to 200. And all the other mineral elements was supplied equally. So this is full view of our facility. So I will present a result from 3 parts. I hope from my presentation we can learn more about the nitrogen effect on the pomegranate. So first is the tree growth. This is a whole view of our experiment. And we start the experiment from 2015, and we have started the different treatment on the February of 2016. And this is well up to the next year, and this is from the 2018, and we have 8 different treatment of the trees. So let's see the detailed result. So we can see for all the parameters, the nitrogen 14 treatment have got the highest growth response. So the canopy volume in 2018 and increased almost 3.4 times compared to 2017. And also for the leaf area density, and they also have the highest value and also the highest increase rate. And also the water consumption increased very significantly from the first year, this treatment from 6 cubic meter to almost 20 cubic meter per tree per year. So we can see the leaf appearance and also the SPAD and leaf area from different nitrogen levels. So the value of the SPAD is increased with the increase of the nitrogen supply, but the area of the leaves is not. We can find there is the highest leaf areas from the 100 treatment, and this can be seen from the following pictures. If we go more closely, and we will find that when the nitrogen supply is excessive, like 200, so the wilting of the leaf tips is more obviously here, and probably due to the reduction of the calcium concentration in the leaves, because the calcium concentration in this leaf is only half compared to this leaf. So from the trunk diameter is all the treatment increase gradually over 3 years. In the first year, the great growth is from the nitrogen 17 and the 100 treatment, but in the third year, the tree of the 14 treatment has the highest growth rate. So optimal concentration changed as the tree grew. And at the end of the experiment, so the nitrogen 5 had the lowest change rate, and the most excessive nitrogen, like 200, has a similar to the 10 treatment. So also in different seasons, like the growth rate is higher in summer compared to the winter. So based on the previous result, we found that there is a significant positive correlation between the trunk diameter changes and also the annual accumulation water consumption. So we assume that trunk variation may set as one of the reference indicators for determining the irrigation in the commercial orchard. And we also, in the fertigation system, mostly when the nitrogen concentration in the irrigation and the drainage is equal, like the 1:1 ratio, so it's be considered to be the most suitable concentration for the tree's growth. So from the correlation between these two parts, we can see the appropriate drainage solution from different growth stage for the trees is also changed. So the appropriate nitrogen concentration also changed for different growth stage. Okay, so the next part is about productivity. So all the fruit is from the flower. So the pomegranate is Andraminaceae plants as disseminate flowers are considered to bear better pollen, like this one, and the hermaphrodites flowers numbers related to the final yield. So we want to know if the nitrogen application may influence the hermaphrodites and to the stamate flower ratio and the relation reproduction parameters to compensate between the two. So from the result, we can see nitrogen deficiency leads to a decrease in a number of the hermaphrodites and staminate flowers per fruit tree. But the nitrogen application change does not affect the fraction of the hermaphrodites flower from total flower, but the fraction of the fruit number from the hermaphrodites flowers. And for the yield, we can see that pomegranate got significantly lower yield in the nitrogen deficient treatment, and the average yield doubled in 2018 compared to in 2017. And the highest yield for individual tree is almost 120 per tree. And compared to the field, this is the first year we have harvest in 2017, but in the second year is already higher than the average in the field. So in this system, we can have very strong productivity ability. Except the yield, the quality of the fruit is also very important and affects the final economy benefits. So in the 2 years, we measured more than 5,000 fruits to evaluate the fruit quality. So the area color, so first we see the fruit appearance quality. So the area color also lighting under the low nitrogen also when it's giving too much. But the fruit black root decrease with the decreasing nitrogen, but the peel got somewhat worse when the nitrogen got lower. But the nitrogen did have any effect on the peel color, cracking, and peel roughness. And for the inside of the fruit quality, we can see the weight, size, and the quantity of pomegranate and areas decrease significantly with lower nitrogen application treatments. And the nitrogen deficiency had a more pronounced effect on the area numbers than the area size. And the nitrogen deficiency significantly decreased the total soluble solid and acid in the pomegranate juice, but did not affect the ratio between them. So both total phenols and flavonoids in the juice were enhanced in the nitrogen deficient situation. So the annual nutrient requirement of the pomegranate trees is achieved maximal yield is around 500 of nitrogen and 100 of phosphorus and 600 gram of potassium. So for this and for all of that, is 1/3 of them need to be supplied in 1 month before the food was harvested. And also the leaf nitrogen increased to a plateau as a function and increase irrigation solution of nitrogen. So maximal at around 15 to 20 milligram per gram of the biomass at a concentration of around 40 and around 40. And the leaf nitrogen is suitable to diagnose the nitrogen deficiency for the pomegranate trees, but is not suitable for the too much of the nitrogen supplies. And another conclusion from my result is that the appropriate fertigation management not only increase the productivity, we can see the green line is have highest productivity of pomegranate fruit, but also improve the fruits and juice quality. And we have less proportion of decayed and fruits and higher concentration of the antioxidant compounds. And therefore to enhance the benefit for both the grower and customers. But because this experiment is conducted in the soilless culture, so we require further step to validate and convert the finding into. We should do this into the horticulture recommendation in the real orchard. So that's all I want to present, and this is some of the paper we published related to the result we already present. And last, I want to use this opportunity to thank the funds from the chief scientist and the CFPN funding, and also to thank a lot of thanks to Anon Uri, Alon Ran, and to give the opportunity for me to study and learn from you all in Israel. And thank you, Sedit, for the preparation for the manuscript we're writing, and also a lot of technical support from the lab member. And also I want to thank Professor Fu Suo and Jian Bo from China Agricultural University to support me to study in Israel. And the pomegranate is one of the very healthy fruits for our body. So I wish everyone will be healthy and safe during such a special occasion. And I'm looking forward to meet you all face to face next time. So that's all. Thank you for your attention.